Hello everyone and welcome. In this video we're checking out variant 2 of October-November 2024, the MCQ. Before starting, make sure to check out the free notes I have available for you in the comment section down below. Now let's go ahead and start with question number 1. A light microscope is used to observe two structures that are 200 nanometers apart on a slide. What's the actual distance between the two structures when the magnification is changed from times 40 to times 400? Okay, he's looking for the actual length and he gave you observe two structures that are 200 nanometers apart on the slide. So he gave you actual length, okay, because he said that they are 200 nanometers apart on the slide. So the real distance between them on the slide is 200 nanometers. Now he's looking for actual distance when the magnification is changed, and actually, heads up, actual length does not change with magnification. It's the image length that changes, and therefore the actual length remains 200. Okay, a cell is shown in the micrograph. Which statement explains how it's possible to identify the type of microscope used to produce a micrograph? Okay, so obviously here we're talking about an electron microscope, because we could see the endoplasmic reticulum, we could see the ribosomes on the endoplasmic reticulum, we could see the membranes of the mitochondrion, we could see a whole lot of things. So it's definitely not an image shown by the light microscope. The nucleus is visible, so it's an electron microscope. No, nucleus is visible whether it's light or electron microscope. The endoplasmic reticulum is not visible, so the light microscope was used. Actually, we can very clearly see the endoplasmic reticulum, which are these folds over here. Okay, so that's also wrong. Okay, chloroplasts are visible, so a light microscope was used. If chloroplast was visible, this could mean it's both either electron or light microscope because it's visible under both. However, I'm not really sure we could see any chloroplast here to begin with, and we did not use a light microscope. Ribosomes are visible, so an electron microscope was used. Exactly. Okay, we could see ribosomes, and ribosomes are very small and require such low or high resolution in order to be seen, which is the case with the electron microscope. Which statement supports the fact that mature plant cells contain organelles that carry out the same role as lysosomes? Okay. Statements support the fact that mature plant cells contain organelles that carry out the same role as lysosomes. Okay, so we know that mature or developing flowering cells contain vacuoles that carry out the function of lysosomes, meaning that in their vacuoles there are hydrolytic enzymes. And so instead of having tiny vesicles, they have the large permanent vacuole containing the hydrolytic enzymes. So a range of hydrolytic enzymes can be found within mature plant vacuoles. Exactly. Glycogen found in vesicles can be hydrolyzed to glucose molecules. Firstly, there is no glycogen in plant cells, and because he did not specify it's a vacuole, so no, this does not show anything. Double membrane bound vesicles are formed from plant Golgi bodies. That's not true. Okay. This is not true. Okay, they do not contain double membrane bound organelles that are formed from the Golgi bodies, and they're not. These double membrane vesicles are definitely not carrying out the same role as lysosomes. Vesicles formed from cell surface membrane contain enzymes. Again, this is wrong. We're looking for the vacuole. Okay, A. Which cell structures may contain cisternae? Cisternae might be confused with a lot of students with cristae. Cristae are the folds in the mitochondrion. So remember, the mitochondrion is going to have two membranes. The outer membrane is nice and smooth. The inner membrane is highly folded. So these are called cristae, so singular crista, okay? Whereas folds in the Golgi body, in endoplasmic reticulum, these are called cisternae. So if we have endoplasmic reticulum connected like so, okay, these folds, they're called cisternae, okay? And so is the case in the Golgi bodies. Here you can see these folds are also called cisternae. Okay, so yes, Golgi body, endoplasmic reticulum, not in the mitochondrion, definitely not in the chloroplast, okay? Chloroplasts have thylakoid membranes, so it's a double membrane organelle, and then within it are these folds of membranes, and these are called thylakoids. So yes, it does not contain cisternae. Okay, so we have check and X, C. Okay, the diagram shows some cell structures of one type of cell. So you have you could see it's a bacterial cell, of course, because we could see the cell wall, we could see the circular DNA, and so on. So which labeled cell structures are present in typical eukaryotic cells and typical bacterial cells? Okay, so one being the plasma membrane is definitely found in both eukaryotic and bacterial cells. Two being a cell wall. And this kind of cell wall for bacteria is made of peptidoglycan, and it's not typically found in eukaryotic cells. So unless they're discussing cell walls in general, then yes, some eukaryotic cells have cell walls. They have cellulose cell walls in plants, chitin in fungi, and so on. So we can get back to 2 based on the answers. 3 is circular DNA. Typical eukaryotic cells do have circular DNA within their mitochondria, within their chloroplast, okay? And 4 being 70 S ribosomes, again, they are found, but just within the chloroplast and mitochondria, okay? And 5 being cytoplasm, definitely it's found within all cells. So 1, 3, 4, 5 is actually our option here, so it's B. If 1, 2, 3, 4, 5, we would have to consider number 2. However, since it's not even considered, so we eliminate it first. Okay. The diagram shows parts of collagen fiber made by collagen triple helices. The collagen triple helices are linked together to each other by one type of bond, and this bond is labeled as X in the diagram. So we have a bond between the collagen fibers, and he says what type of bond is bond X. Well, bond X is actually a covalent bond, and so this is going to be A. So please don't confuse how collagen fibers link up with how cellulose fibers link, because cellulose fibers only bond with hydrogen bonds, whereas for collagen it's going to be covalent bonds. The table shows some information about polypeptides and make up hemoglobin. So there's alpha globin, there's beta globin, and the total number of amino acid residues in the polypeptide chain. So you could see a total number of amino acid residues in the polypeptide chain. So this is a little bit longer, and the position of the amino acid cysteine in the polypeptide chain, here it's at 104. Here we have 2 positions for the amino acid cysteine. So let's remember together the structure of hemoglobin. Hemoglobin is made of two different chains, and it's total made of 4 polypeptides. Okay, so we have this kind of thing over here, and then again like this. So these are 2 chains, and then we also have 2 different chains. So these orange ones, they're going to be the beta chains, and then the purple ones are going to be the alpha chains. Okay, now so here he says that the alpha chains are a bit longer and a bit shorter, and that they contain amino acid cysteine at position of 104. So that's right here. Maybe they contain cysteine. And remember, cysteine is the amino acid that forms the disulfide bonds because it contains the R group, which is SH. Okay. Let's see what they're looking for in this question. Scientists studied in the region of beta-globin polypeptide chain containing the amino acid cysteine at position 93. They found that the region faces outwards with no oxygens attached to the heme group. Okay, so the region... they said basically they studied the region of peptidoglycan chain containing the amino acid cysteine in position 93, and they said that this region faces outwards with no oxygen attached to the heme group. Okay, so when no oxygen is attached to the heme group, this position of 93, okay, it's facing outwards. I think once oxygen attaches to the heme group, it's a bit shifted, yes? So the region faces inwards when oxygen is attached to the heme group. Now this is known as cooperative binding. When oxygen binds, it's going to change the shape of hemoglobin a little bit so that it makes it easier for the next oxygen to bind to the next heme group, and so on. Okay, now I don't really see how this relates to cysteine, but we're gonna get to the point, I guess. Replacing cysteine with a different amino acid reduces the Bohr shift. Okay, so that's a point he's saying here, that if you replace cysteine with a different amino acid, you're reducing the Bohr shift. I guess it has to do with the kind of bonding that's going to happen when maybe cysteine goes inwards instead, so that maybe it's able to form disulfide bonds. Let's see what he says here. What can be concluded from the information about cysteine in hemoglobin? More than 1% of amino acids in one hemoglobin protein are cysteine. Okay, from this information we can't really conclude this, but from the table we totally can. He says here that there's a total number of 141 in alpha chain, 146 in beta globin. So 141 plus 146 is a total of 200, and obviously we're going to have to multiply each by 2 because there's 2 chains in each, and so we get a total of 574 amino acids. And remember, I multiplied by 2 because there are 4 chains, not just 1, so there's 2 of each chain. And then he says here that we only have 1 amino acid here, so there's 2 amino acids in total alpha chains, and then there's also 2 amino acids here, so 4 amino acids total in all beta chains. There's 2 here and 2 here, so that's 4. So 2 plus 4, that's 6. So now 6 divided by 574 is equal to, and then multiply of course by 100 because it's a percentage, and yes, we get approximately 1%. So and it's actually a little more than 1%, so yes, A is correct so far. Let's check out the other options. In alpha globin there are in alpha globin there's a cysteine closer to the end of the polypeptide chain with an unreacted carboxyl group than beta globin. Okay, interesting. So you're saying here that in alpha chain, a cysteine is closer to the end of the polypeptide chain with an unreacted carboxyl group than in the beta globin. Okay, so in the beta globin we have two what's it called, cysteine atoms on each, and there is obviously the end that has the COOH and then the end that has the NH for each amino acid. Yes. Now the fact that the alpha globin, the cysteine is closer to the end versus this guy, well, actually you'll find that alpha globin, okay, like if this is a like valid question, then these numbers do not really work, because the alpha globin has cysteine in the middle. Yes. So if, let's say, the C end is right here and the NH end is right here, so COOH is right here and NH end is right there, then this guy is going to be closer. I mean, beta globin is going to be closer because the C or cysteine is in the middle. Okay, you'll find that the position is 104, whereas here we have two positions, 93 and 112. So we have above it and we have below it. So either, in which case, like either scenario, okay, whereas like maybe NH2 is here and COH is here, doesn't matter. Either way, beta globin is going to be closer because it has two options. It could be above or it could be below, based on obviously where the COH end is present, which right now I don't think we can locate. Okay, and we shouldn't be wasting a lot of time on this question because no, the question itself is not valid. Okay, moving on to C. The replacement of the cysteine at position 93 in beta globin decreases the affinity of hemoglobin for oxygen at low pH. Okay. At low pH, okay, if you replace cysteine at position 93 in beta globin, that decreases the affinity of hemoglobin for oxygen. Now, replacing cysteine with a different amino acid will reduce the Bohr shift, which means that cysteine, if it's replaced, this is going to actually, yes, it's going to decrease the affinity of hemoglobin for oxygen, but the specification of low pH is a little bit tricky here. I don't get why it's going to decrease the affinity at low pH, where like, the replacement of cysteine at position 93 in beta globin decreases the affinity of hemoglobin for oxygen at low pH. I think that again this is wrong, because here he refers to multiple points and none of which like apply to what is said upwards. He said here replacing cysteine with different amino acids reduces the Bohr shift, and that is true, which means if you replace it, this is going to change the affinity of hemoglobin to oxygen. However, at low pH, this is dictated by the carbon dioxide concentration. It has nothing to do with cysteine. The binding of oxygen to the heme group causes the region of beta globin containing cysteine at position 93 to become more hydrophilic. Okay, the binding of oxygen causes the region of beta globin containing cysteine to become more hydrophilic. Now, he said here that the region faces inwards when oxygen is attached to the heme group. So I don't think it's becoming more hydrophilic, because if it faces inwards, this means that it's running away from the water, which is outwards. And so I don't think it's becoming more hydrophilic. I think it's A. Okay, which features of glycogen distinguish it from starch? So, distinguishing it from starch, all glycogen molecules are highly branched. Okay, starch is made of two molecules. It's made of aminopectin and amylose. Aminopectin is highly branched, whereas amylose is not branched at all. And so, yes, all glycogen molecules are highly branched, and this is a distinguisher between starch, which has one molecule that is absolutely not branched. So, A is right. All glycogen molecules are polysaccharides, well, so are all the molecules from starch. All glycogen molecules contain alpha glucose, well, so are all the molecules from starch. All glycogen molecules contain 1,4-glycosidic bonds, well, so are all the molecules from starch. The diagram shows a biological molecule. So we have a biological molecule, and we can see a triglyceride, we can see glycerol and 3 fatty acid chains. Which molecules would be produced if this biological molecule was hydrolyzed? If we hydrolyze it, we're going to produce glycerol and 3 fatty acids. Now here, from the options, we have amino acids. Obviously not. We're not releasing any amino acids here. Fatty acids and glycerol, or fatty acids, glycerol, and water. Remember, to hydrolyze means to use water. You're not going to be releasing water when you are using it, and that's why this is wrong. And so it's just fatty acids and glycerol. Okay, if it was a condensation reaction, like in the formation we're going to release water. But in the breaking down, we're going to use water to break the bond, and so no water is released, water is used. Okay, moving on to question 10. A mixture of glucose and starch solutions was placed in a length of dialysis whisking tubing, and the tubing was sealed, and the tubing was then placed in a boiling tube containing distilled water, and 2 samples were placed immediately. Removed this water and tested with either iodine solution or Benedict solution, and this was repeated at 10-minute intervals for 30 minutes. Okay, interesting. And the iodine solution gave an orange-brown color each time. There was no change because remember, if you have a dialysis tubing bag and it contains starch and glucose, glucose is capable of leaving the dialysis tubing bag, whereas starch is too large to leave the dialysis tubing bag. And so starch is going to stay inside and iodine is going to come react to it, whereas glucose is going to leave and then it's going to react with Benedict's solution. So the tube shows the results for Benedict's, and you can see how with time the concentration of glucose increases in the solution because you can see how the colors get more and more concentrated as glucose diffuses outside and it starts to react with the Benedict's solution. Now, what may be concluded from these results? So what is concluded from the results is that the pores in the Whisking tubing are too small for starch to pass through. That is right, because you can see it remains an orange-brown color each time, which means that the test is negative, and so starch could not leave, and so it, because obviously we're testing the water, we're not testing inside the dialysis tubing bag, there is no reaction, and so this is why the pores, that is true. Okay, glucose diffuses from the Whisking tubing down a diffusion gradient, exactly, which is why the concentration keeps increasing after time. Water diffuses into the Whisking tube. Actually, this is definitely not concluded by the reaction, and because glucose is leaving, I don't see why water would be moving in. So no, this is not concluded by the results. It's 1 and 2 only, which is B. Which molecules are globular proteins? Globular proteins are hydrophilic ball-shaped molecules with tertiary structures. So if they're an enzyme, they're definitely globular. Let's see the options. Amylase is an enzyme, it's globular. Hemoglobin is not an enzyme, however, it is a protein that has a tertiary structure. It is hydrophilic because obviously it lives within the red blood cells cytoplasm. And cytoplasm is 70% water, so it's definitely hydrophilic, and it's definitely ball-shaped, so 2 is also globular. DNA polymerase is also globular, it's an enzyme. 1, 2, and 3 are all globular proteins. An initial rate of reaction catalyzed by an enzyme was measured at various substrate concentrations. Which graph shows the effect of a low concentration of non-competitive inhibitor on the reaction? Okay, non-competitive inhibitor, remember the graphs of results. So we have different graphs. We have the graph in the case of a competitive inhibitor, and the graph in the case it's normal, and the graph in the case of non-competitive inhibitor. And in all graphs, we're testing against substrate concentration. So this is non-competitive inhibitor. So in the case it's normal, the rate of reaction, keeps on increasing as the substrate concentration increases until, we reach a point where it's no longer a limiting factor and we reach a plateau. Whereas with competitive inhibitor, what happens is that let's just combine them together. So this is basically the normal graph, okay? Now with a competitive inhibitor, it's gonna be something like this. Okay, it's like this, okay? So you can see how it takes time for the enzyme to reach its maximum rate of reaction because there's something blocking it, because the competitive inhibitor blocks it, because this is the enzyme, and the enzyme has an active site, and the competitive inhibitor is awfully similar to the substrate in shape, and so it stops the enzyme from reacting with the substrate. Instead of reacting with the substrate, it's reacting with the competitive inhibitor, basically wasting time. Whereas for the non-competitive inhibitor, let's draw the normal graph, and now let's draw it with the non-competitive inhibitor. It's going to be something like this, okay, where you can see the enzyme never reaches the Vmax or maximum rate of reaction, and it always stays here, it plateaus, and that is because the non-competitive inhibitor is going to stop the enzyme from functioning by binding to another site called the allosteric site, not the active site. And so once the non-competitive inhibitor binds to this allosteric site, the enzyme loses its active site, and so what happens is it can no longer catalyze the reaction, and so the rate of reaction never increases. Okay, so you're going to find that if we have this graph, that's going to be the answer, and which is basically D. Okay, moving on to 13. Gout is a type of arthritis in which small uric acid crystals form inside and around the joints. It causes sudden attacks of severe pain and swelling, and the diagram shows how uric acid is formed from hypoxanthine catalyzed by enzyme xanthine oxidase. Okay, so hypoxanthine is going to turn into xanthine and turn into uric acid, all by the actions of xanthine oxidase. Now, he says here, gout can be treated using a drug called allopurinol, which has similar shape to hypoxanthine. So it has similar shape to the substrate, which is basically how competitive inhibition works. The competitive inhibitor has to have a similar shape to the substrate so that it blocks the active site of the enzyme and basically stays there because it has a very similar shape to the substrate, and hence it's complementary to the active site, and so obviously reducing the rate of reaction. What can be concluded from this information about how allopurinol prevents the formation of uric acid? Again, let's repeat this. This is allopurinol, and then this is the substrate, and this over here is the enzyme. Okay, now this is enzyme, and this is substrate, and this is the inhibitor or competitive inhibitor. You can see how the competitive inhibitor is awfully similar to the substrate in binding site, and so instead of the enzyme binding to the substrate, it thinks that this is the substrate and it binds to it, and when it binds to the competitive inhibitor, this basically delays the reaction and it reduces the rate of reaction. Okay, now you could see here what could be concluded is it binds to the active site of xanthine oxidase instead of hypoxanthine, resulting in reduced production of uric acid. Exactly, and that's why it's A. It binds to another part of xanthine oxidase, and this changes the shape of the active site. This is wrong. This is how non-competitive inhibitors work. They bind to a different site other than the active site called the allosteric site, and then they change the shape of the active site, hence reducing the rate of reaction. But this is not what happens here, because he told you specifically from this information, which is the fact that it has a similar shape to hypoxanthine, this means it's a competitive inhibitor. It disrupts the hydrogen bonds with xanthine oxidase, so it denatures, and the active site is no longer complementary to hypoxanthine and xanthine. No, because if it denatures, this is the action of things like different chemicals or things like the pH or temperature, but definitely not due to this chemical, especially since they didn't list any information about this chemical of like its capability to do so. So this is wrong. It hydrolyzes the peptide bond with xanthine oxidase to change the shape of the active site. Of course, if it's a protease enzyme and if they listed that, I would have chosen this, but again, this is wrong, and so A is the answer based on the information provided. 14. Phospholipids are formed in a similar way to triglycerides. Yes, so it's formed to a similar way to triglycerides, and a sample containing 6 phospholipid molecules. Okay, so we have phospholipids. What is the structure of phospholipids? We're going to have a glycerol molecule, and the glycerol here is going to be attached to a phosphate group, and it's going to be attached to 2 fatty acid chains. So yes, it's awfully similar to triglycerides in that triglycerides are, instead of the phosphate group over here, it's attached to a third fatty acid chain. So a sample contains 6 phospholipid molecules, and the molecular weight of phosphate ion was 95. The molecular weight of each individual fatty acid was found to be 282. The molecular weight of glycerol is 92. Molecular weight of water is 18, and he's looking for the molecular weight of the sample, which, let's remember, the sample contained 6 phospholipid molecules. So whatever the mass of the phospholipid comes out to be after we found this information, we need to multiply the final answer by 6. Okay. Let's see. Firstly, the mass of the phospholipid, we should take into account this information over here. It contains 1 phosphate group. Okay, it contains 2 fatty acids. It contains 1 glycerol. And let's not forget how each bond is formed. It's formed by condensation. Condensation means the removal of water. So the bond here between glycerol and phosphate is formed by condensation. The bond here between glycerol and the fatty acid is formed by condensation. And also the bond here between glycerol and the fatty acid chain. Now, I think this might be confusing to someone who might, for example, not notice that this is a bond formed by condensation. And I will say to you, let's together remember the structure of glycerol. Glycerol is basically C connected to OH, 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 and then a whole bunch of H's. Now, this means that in order to basically bond with anything, so if anything wants to bond to it, for example, like a fatty acid, what happens was a condensation reaction. So here we're going to have the formation of water, and then same thing here with the formation of water with a different fatty acid chain. And lastly, if a phosphate group comes in with its different oxygen, and like H here, it's going to be able to do condensation all over again. So what I'm trying to say here is that don't forget the water removed in this bond. So let's go ahead and calculate. We're going to have 1 phosphate, that's 95, 2 fatty acids, that's 282 times 2, and what's it called, 1 glycerol, so that's 92, and then minus 3 water molecules, so that's 3 times 18, and this is gonna give me, let's see, 95 plus 282 times 2 plus 92 minus 3 times 18, and that's going to be 697. Now we're gonna multiply the answer by 6, and this should give me 4182, which is B. Moving on to 15, which statement about phospholipids and cell surface membranes are correct? Fatty acid tails allow most ions to pass through the membrane. Hydrophobic tails point inwards towards each other. All polar heads face the cytoplasm. The phospholipids help with the flexibility of the membrane. Okay, so let's see. Again, how the structure of the membrane looks is basically a whole bunch of phosphate heads, a whole bunch of fatty acid chains, and the fatty acid chains are facing inwards towards each other because they're hydrophobic. And then on the opposite sides of the membrane, we're going to have some fluid over here. So we have, for example, the cytoplasm, which is basically made of water, 70% water, and then on the other surface we have extracellular fluid, which is again mostly made of water. So information number 1, fatty acid tails allow most ions to pass through the membrane. That's definitely wrong because fatty acids are hydrophobic, ions are hydrophilic, they can't pass through the membrane so easily, they need a carrier. 2, hydrophobic tails point inwards facing each other. This is true. All polar heads face the cytoplasm. This is wrong because some polar heads, as you can see here, face the cytoplasm. Some polar heads face the extracellular fluid, so that's wrong. The phospholipids help with the flexibility of the membrane. Yes, they do, the phospholipids, depending on the shape and the type of the fatty acid chain, that's going to determine the flexibility or fluidity of the membrane. And so this is true. So it's 2 and 4 only, which is D. In an experiment, pieces of onion epidermis are put into 3 different concentrations of sucrose solutions, P, Q, R, and the pieces of onions are left for an hour and then examined under the low power of a light microscope. Now each diagram shows the cell from epidermis that was placed in each sucrose concentration. You could see how P, Q, and R, you could see how, for example, in P it's turgid, and that's probably because it's placed in something like distilled water. In Q, we can see it's a bit flaccid, which means it's placed in isotonic solution, so the concentration or the water potential outside equals the water potential inside. R is, as you can see, plasmolized, so it's placed in a hypertonic solution, something really concentrated. So let's see what explains the appearance of solution Q. The concentration of solution Q is equal to the concentration of solutes in the cell sap. Okay, so the concentration of solution Q is equal to the concentration of the solutes in the cell sap. The cytoplasm has the same concentration of sucrose as solution Q. So the cytoplasm having the same concentration of sucrose specifically is really not the answer, because obviously sucrose is just one thing, whereas inside we have a whole bunch of other molecules. The water potential of the cytoplasm is equal to the water potential of the vacuole. Okay, that's a bit confusing. Why would we care about the vacuole? Okay, the vacuole and cytoplasm, the osmosis or diffusion is going to happen here, whereas we're looking for what happens between outside and inside, and that's going to be equilibrium. Water potential of the cell sap is equal to the water potential of solution Q. This is what I'm looking for. Osmosis is the reason that the shape of the cell is changing, and that's why I'm looking for water potential specifically, where water potential is equal to the water potential outside because it's an isotonic solution. Okay, 17, the electron micrograph shows rod-shaped bacteria, and the actual length of the bacterium is 2 micrometers and the diameter is 0.5. So he gave you the length of the thing and he also gave you the diameter, and assume the bacterium is cylindrical in shape. So he's asking us to assume it's like this, like a cylinder, instead of like this shape. So he says here, what's the surface area to volume ratio of the bacterium? Okay, surface area to volume ratio could be calculated by first getting the surface area, second getting the volume, third dividing them by each other. Okay, so first things to do, the actual length of bacterium. So first we need to calculate the surface area. Now surface area could be calculated by the following. We get the surfaces of the circles here, and we add them to the area of the curved region of the cylinder. Okay, so the area of one circle is pi r squared. Okay, so we have 2 circles, so it's 2 pi r squared. The area of the curved region over here is going to be the area of the circumference of the circle, pi r, times the length here, so pi r l. Yes, as I mentioned, it's the circumference, so it's not just pi r, it's 2 pi r, because 2 times r is the diameter. Anyway, it's a mathematical formula. I need you guys to memorize it. please. Okay, now let's go ahead and figure out what the volume is. So the volume is a quite famous formula. It's pi r squared h. So we're gonna get back to this. Let's first calculate the surface area. So surface area is going to be 2 times pi times, now he gave you the length and he also gave me the diameter, but I'm looking for radius. Radius is equal to half the diameter. So radius is equal to 0.5 divided by 2, which is 0.25. Okay, so 2 times pi times 0.25 squared plus 2 times pi times 0.25 times the length, which is 2. And so this is going to be so this gave me 9 over 8 pi, whereas the pi r squared h here is going to be equal to pi times 0.25 squared times the height of 2. So let's figure this out. And that's going to be 1 over 8 pi. So 1 over 8 pi, and we have 9 over 8 pi. So if we divide them together, 9 over 8 pi over 1 over 8 pi, 8 cancels, pi cancels, and we get 9 over 1, which is a ratio of 9 to 1, which is D. Chickens have 78 chromosomes in the nucleus of the body cell. How many DNA molecules are there in the chicken body cell at the start of prophase of mitosis? Remember, DNA molecules is different to chromosomes. If he asked for chromosomes, it would be the same number, because this is mitosis, which is going to be equational cell division. So DNA molecules is different, because DNA molecules is the same as chromatids. Okay, so at the start of during the S phase, there's going to be replication. During G1, we just had cells with a nucleus with a single chromatid chromosome with a single chromatid. After the S phase, we had a single chromosome but with 2 chromatid joined together at the centromere. And then at the start of prophase, it's going to be the same as S phase, so it's going to be twice as much, 78 times 2, and that's going to be 156. D. The photomicrograph shows cells undergoing mitosis. So you have mitosis, and you can see we have here X. Which statement would describe what happens next in the cell? Okay, sure. So you can see here that chromosomes are very condensed. I think this is just prophase has just happened. There's no nucleus, and the chromosomes are still like at the center of the cell where like the nucleus was. I think now we're going to have metaphase after this, because it's PMAT, prophase, metaphase, anaphase, and then telophase. So after this we need metaphase. He says here chromatin coil up tightly and the nuclear envelope breaks down. This is, I think, what just happened. This is prophase. Okay, chromosomes line up at the equator of the cell to attach the spindle exactly. This is what happens in metaphase. Chromosomes now are going to line up at the equatorial plate and they're going to attach to spindle fibers. Sister chromatids move toward opposite poles pulled by spindle fibers. This is actually anaphase, and no, I think the second step is metaphase. Okay, spindle fibers break down and the cell prepares for cytokinesis. This is telophase. Again, the step we're looking for is metaphase, which is B. Which process occur in bone marrow cells that are mitotic cell cycle? Okay, so in the bone marrow cells, in a mitotic cell cycle, the phosphate groups bind to ADP to form ATP. Okay, respiration, we have formation of ATP. Okay, it could happen as well. Okay, why not? Okay, bonds form between nucleotides and a DNA strand. Exactly, there's going to be DNA replication. Hydrogen bonds form between tRNA anticodons and mRNA codons. Yes, formation of proteins, formation of enzymes, formation of organelles is going to happen during the first and second growth periods, which is why you're going to find protein synthesis. tRNA anticodons and mRNA codons is translation, which is a part of protein synthesis. 1, 2, and 3A. Now, the enzyme telomerase prevents loss of telomeres after many mitotic cell cycles. Which cells need to transcribe telomerase enzyme? Okay, stem cells definitely need to transcribe telomerase. If you don't know what telomerase is, you have in a chromosome, so this is a chromosome, okay, you're going to have these ends which are non-coding. Now these ends are called telomeres. Okay, these telomeres protect your DNA. So you have DNA all the way from here to there and everything, and then there's the ends that code for nothing. And this is because DNA polymerase, when it comes to code for something, when it comes to replicate something, it can't go until the very end. It can't start from the very beginning. And so what happens is that it does not do that. Okay, it deletes a part of the DNA. So it's better for your body to have it delete the part that is not coding for anything than for it to not replicate but without like an important part, which is obviously wrong because it's a damaging of the DNA. And that's why stem cells, they keep the telomerase, because this enzyme is going to elongate the telomeres, protect the telomeres, which is obviously important in DNA replication. And there's going to be lots of DNA replication in stem cells since they do a lot of mitosis. They are constantly replicating. And remember, a part of replication is the S phase, is the S synthesis phase, DNA replication. So stem cells definitely. Activated memory B lymphocytes also divide a lot. They produce plasma cells, and so since they divide quite a lot, we're actually going to need some telomerase here. Helper T lymphocytes secreting cytokines are just doing protein synthesis. They're not going to require telomerase enzyme. 1 and 2 only, which is B. A polypeptide molecule contains the amino acid sequence glycine, leucine, lysine, valine. The table shows DNA triplets for these amino acids. So there's glycine, CCC, leucine, GAA, lysine, TTT, and valine, CAA. And he's saying which tRNA anticodons are needed for the synthesis of this polypeptide. Okay, this question I think a lot of people get confused in, so my advice for you is draw. Okay, he gave you the DNA and he's looking for the tRNA anticodons. Now, since he gave you the DNA, remember this is DNA that's going to be copied. So this is the template strand. Now, template strand, okay, this is template. So template is going to be copied and turned into mRNA. And remember, mRNA is complementary to the template strand. Okay, now mRNA is going to be translated and turned into, and translated by tRNA. Yes, so tRNA is complementary to mRNA. Now we're looking for tRNA and we have the template strand. So since tRNA is complementary to mRNA is complementary to template, that means that tRNA and template are basically the same. Okay, with obviously the exception that DNA does not contain the nucleotide uracil, and so, I'm sorry, not the nucleotide, the nitrogenase base uracil, and so any T's will be turned into U's. Okay? So here it's going to be exactly the same, except that TTT here is turned into UUU. CCC, GAA, UUU, CAA, which is B. The diagram shows a section of a strand of DNA. He's saying what type of bond is X. So this is the phosphodiester bond, and it's called phosphodiester because we have a phosphate group forming a carbon-oxygen bond, carbon, and here a phosphate. Remember, a phosphate group is consistent of POOH. Okay, so it's a carbon-oxygen bond between, which happens twice, okay, with a phosphate group. So it's called a phosphodiester bond because this CO bond is the ester bond. So it's definitely D. Moving on to the 24. Which row correctly describes cytosine? Ring structure, double. Okay, cytosine is a pyrimidine. Pyrimidines have a single ring, and so another example of a pyrimidine would be thymine, whereas the other purines are actually adenine and guanine. So cytosine is going to have a single ring structure. How many bonds does it form with the complementary base? Cytosine is complementary to guanine and it forms 3 hydrogen bonds with it. So it's 3 type of base. So type of base, oh yes, okay, ring structure, type of base is pyrimidine, so that's C. Okay, during the production of protein molecules, only one strand of DNA double helix is used. Which name is given to the DNA strand that's used to produce a new protein? To produce the new protein is going to be the template strand. The template strand is going to be copied or transcribed really by the transcribed during transcription to form mRNA, and then mRNA is used and translated with tRNA, and so it's definitely the template strand. The diagram shows a longitudinal section of the phloem sieve tube and with a companion cell. Where are the mitochondria located for the release of energy for co-transport? Okay, so mitochondria located to release energy, they're definitely not going to be in the sieve tube element. They're not going to be, because remember, sieve tube element is hollow inside, and yes, there's a little bit of cytoplasm on like the ends. It's not going to be there in the middle of the sieve tube element. Okay, so A is wrong. B, yes, definitely it's going to be in the companion cell providing energy for what's it called, translocation. C, on the cell membrane. I don't think there are any mitochondria on the cell membrane. D, on the end plates. Definitely not. There is nothing at the end plates except for like obviously these end plates. Okay, 27. The diagram shows a phloem sieve tube element and a companion cell that are involved in translocation of sucrose. So which process correctly describes the translocation of sucrose through these cells? Okay, Let's remember how translocation happens. So this is the companion cell, and this is the sieve tube element. Now what happens is that first we're going to have hydrogen ions in the companion cell, and they're going to be pumped from Y to Z. So they're going to undergo active transport, and they're going to be carried by proton pumps. And then there's going to be an increasing concentration in the hydrogen ions here. So hydrogen ions are going to undergo basically facilitated diffusion. So they're going to go through a channel protein. Okay, and when they do this, okay, because they have a lot of energy from potential energy from being a lot on one side and low on the other side, what happens is this energy allows them to co-transport with them the assimilate, which is going to be sucrose or amino acids or whatever. And so sucrose is going to be carried with hydrogen ions via indirect active transport. Okay, so the entire process is called indirect active transport. And then that's it. Sucrose is going to now move into the companion cell and then move through the companion cell into the sieve tube element, and mass flow is going to happen. Which process correctly describes the translocation of sucrose through these cells? Sucrose moves from Y to Z by active transport. No, it does not move in this direction. Okay, protons move from Y to Z by active transport. That is true. Okay, protons move from Z to Y by diffusion. By facilitated diffusion. And I don't think they didn't go into specificities here. Protons move from X to W by diffusion. From X to W, protons do not move from X to W by diffusion, so it's definitely B. Which type of molecules are co-transported into companion cells? Sucrose, amino acids, that kind of thing. Monomers and disaccharides, true. Okay, monomers, amino acids, disaccharides, sucrose. Monomers and polysaccharides, no, there are no polysaccharides being translocated. Polysaccharides stay within the cell, within the storage compartments, like for example, starch stays within chloroplast. Okay, polymers and disaccharides, no, again, no polymers, so it's definitely A. Okay, the diagram shows a section through the human heart. Which label is correct? Okay, labeling. Let's see if you can label. Pulmonary artery. This is definitely not the pulmonary artery. This is the aorta. Okay, let's see. What do you think left ventricle is? B is the left ventricle. Is B the left ventricle? No, actually B is the left atrium. Okay, so remember the upper chamber is the atria, whereas the lower chambers are the ventricles. Okay, C is the right atrium. That is actually correct. Okay, it's the upper chamber on the right side. It's the right atrium. Okay, what about D? D is the pulmonary artery. It's again labeled wrong, therefore C is your answer. The graph shows how the volume of the left ventricle changes during one cardiac cycle. Which point on the graph represents the start of atrial systole? The start of atrial systole. Let's see. Firstly, remember there are three phases in the contraction of the heart. It's going to be atrial systole, ventricular systole, and the generalized diastole. Now, during generalized diastole, blood is going to be collected. Then after that in systole, there's going to be pumping of the blood. Okay, so in atrial systole, the blood is pumped from the atria to the ventricles. In ventricular systole, blood is pumped from the ventricles to the entire body. In generalized diastole, there's going to be collection of blood by both the atria and ventricles. So what happens here is we can see there is a decrease and a sharp drop in the ventricular volume, and I think it's because of ventricular systole. So here we had ventricular systole. After ventricular systole, blood starts to collect, and this is going to be the generalized diastole phase because it's ventricular systole, then generalized diastole, then atrial systole, and then we repeat the process all over again. Remember, it's a cycle, so you can start it wherever you like. Okay, so you can imagine, yes, blood is collecting here, and then after enough blood is collected in the atria, they push more of this blood to the ventricles, then ventricles push all the blood at once to the entire body, and so I think this is the start of atrial systole. Okay, so it's not B because B is going to be where, okay, yes, blood is going in. So we might think, okay, but wouldn't like the atria push the blood in? No, blood is going to enter from everywhere. Okay, it's going to enter in the atria, it's going to enter in the ventricles. Then atria contract to push the blood immediately to the ventricles, which is basically what happens here. So there's a sudden increase in volume here. And then ventricles are going to push all the blood out, and this is what's going to happen. Moving on to 31, which statement is correct? All our blood cells, in our blood cell, CO2 can combine with hemoglobin to form hemoglobinic acid. Wrong, hemoglobinic acid forms from hemoglobin combining with hydrogen ions, so A is wrong. Carbonic anhydrase is the enzyme that catalyzes the reaction between CO2 and H2O. This is correct. Okay, at the lungs, carbon dioxide is released when carbonic acid and water react together. No, actually what happens is that carbonic acid is going to break down into CO2 and H2O. So carbonic acid breaks down into carbon dioxide, and water. Okay, carbonic acid does not react with water. It releases water. So this is wrong. Okay, the greater the concentration of CO2 in the blood, the higher the affinity of hemoglobin for oxygen. Actually, no. If you increase the concentration of hydrogen ions, carbon dioxide, carbon monoxide, these all are going to decrease the affinity of hemoglobin for oxygen. The only way to increase the affinity of hemoglobin for oxygen is to increase the concentration of oxygen. So this is also wrong, and so the answer is B. The diagram, or which row, is correct for an artery? 32. Okay, inner layer of an artery contains squamous cells, so smooth layer of squamous cells. Middle layer, collagen, elastic fibers, and smooth muscles. Outer layer, collagen and elastic fibers. Okay, middle layer is going to contain actually a little bit of collagen. Yes, okay, you could say that it does contain collagen. And then the outer layer is going to contain mostly just collagen. There's no muscles in the outer layer, so C is my answer. 33. Which row shows the features of gas exchange surface that increase diffusion of carbon dioxide and oxygen? Concentration gradient should be steep to increase the diffusion. Diffusion distance should be short. Okay, so it's just a basic question about diffusion, steep and short. Moving on to 34, the diagram or the electron micrograph shows some of the airways in the gaseous exchange system of an insect and respiring body of cells that surround them. So you have a respiring body, and you have each trachea is filled with air, which enters through tiny holes called spiracles. Each trachea is held open by spirals of materials called chitin, and tracheoles are narrow tubes which branch from a wide trachea, and they lie next to the body, and gas exchange occurs across them directly into the respiring body cells. Which statement describes correct differences between the insect gas exchange system shown in the electron micrograph and the human gas exchange system? Okay, gas exchange occurs through the walls of the airways directly into the respiring body cells in insects, but that does not happen in humans. It is true, and exchange for us is going to require a transport system. So after at the lungs there's going to be exchange of CO2 and oxygen. This oxygen is going to be carried by blood pumped from the heart through your blood vessels and everything. It's not going to be directly with your body cells because we have a great surface area to volume ratio. We are huge compared to the tiny insects that can't afford to do this. Okay, they can do it quickly. We have lots of cells in us, and so it cannot really happen. Okay, there are spirals of chitin in the walls of trachea in insects to hold it open, but not in humans. That is actually true. Okay, for us, trachea is held open by the C-shaped rings of cartilage, whereas in insects, spirals of chitin. Okay, there is more than one trachea in the gas exchange system of insects, only one trachea in humans. It is true, they actually labeled here each trachea, and we only have one trachea in the human body, so it's 1, 2, and 3. A. Which row is correct for the wall of trachea and the wall of the bronchus? Cartilage, smooth muscle, goblet cells are found in both equally, so that's A. Moving on to 36, a bacterial pathogen produces a protein that acts as a toxin. The toxin is harmful to humans, and scientists are developing monoclonal antibodies that can be used to detect the presence of toxin in the body so that early treatment can be given. Which statements describe steps in the development of these monoclonal antibodies? Let's review monoclonal antibodies together. So monoclonal antibodies are these antibodies produced outside of the body by some other let's say animals. For example, a mouse is going to be used in the lab. So this mouse is very sad. It's going to be injected with a pathogen. So we're going to inject a pathogen over here, and this pathogen is going to obviously stimulate a primary immune response, which results in the production of B memory cells, or just going to result in B memory cells producing these plasma cells that are going to obviously fight against the pathogen by releasing antibodies. So these are the plasma cells produced by the mouse's body. After this, what we do is we take the spleen of the mouse, because the spleen is basically a part of the lymphatic system, and so we take the spleen, we basically mix it and everything. Okay, it sounds like animal abuse, but trust me, it's what happens is that basically we're going to get the plasma cells, and basically all the plasma cells that are in the mouse's body. So once we get all the plasma cells from the mouse, we're going to place them on trays. No, actually, after we get the plasma cells, sorry, I skipped a step. Yes, after we get the plasma cells, we're going to fuse them with cancer cells, which are the myeloma cells. Now, once you fuse them together, you get a hybridoma cell. This hybridoma cell has two properties. It can produce antibodies, plus it can replicate really fast, which means it allows us to achieve our goals here of getting a whole bunch of antibodies. And this basically is where we get the tray with the wells, and we start separating the cells, the hybridoma cells. So each hybridoma cell is placed in a well, because each hybridoma cell is basically a cell that can produce specific kinds of antibodies, because it's made from different plasma cells. And all these plasma cells were taken from the mouse, and the mouse, it's not only faced this pathogen before, it's faced a whole bunch of pathogens in its life. And we're looking for the specific hybridoma cell that we want, okay, the specific pathogen that we injected. So we keep on putting them in the wells to identify, oh, this is actually the one, and then we take it and we get it to replicate, producing a whole lot of the antibodies that we want. Which statements are correct for penicillin? Oh, okay. So the toxin, number 1, toxin protein is injected into the mouse and triggers the mitosis of specific B lymphocyte. Exactly. Antibodies are collected from the spleen of the mouse and fused with myeloma cells. That is actually wrong. It's not the antibodies that are fused with the myeloma cells, it's the plasma cells from the B lymphocytes. So 2 is wrong. 3, a hybridoma cell produces many antibodies with different variable regions. Actually, no. He says here that it's a hybridoma cell, just one hybridoma cell, produces many antibodies with a variety of different variable regions. The variable regions are going to be the antibody binding site of the antibody. And so, for the hybridoma cell to produce different kinds of antibodies is quite weird, because it's supposed to produce a specific kind of antibody, which is B. Which statements are correct for penicillin? Penicillin, it is harmful to prokaryotic cells. Penicillin is an antibiotic, so yes, it's definitely harmful to prokaryotic cells and for their growth and development. It disrupts cell wall synthesis, exactly. Penicillin is going to stop the cell wall, specifically the fibers of the peptidoglycan, from forming bonds with each other. And so then when the wall tries to grow, it's not going to be able to grow anymore, because there are no bonds between the different units of peptidoglycan, and it's eventually going to burst and everything. We're not going to get into detail here, but it becomes less effective with regular use. Actually, it becomes less effective with regular use. I mean, you can apply that to the fact that regular use results in antibiotic resistance, and so yes, it does become less effective with regular use, but that's not because the penicillin itself is becoming ineffective. So yes, I'm gonna pick 1, 2, and 3. Why is passive immunity for only a short term? Passive immunity is only for a short term. Examples of passive immunity would be just providing someone with antibodies. It's because the person here does not produce any memory cells. They do not even undergo primary or secondary immune responses. They are just using the antibodies given to them to fight the disease. Now in some other case where we're talking about active immunity, in that situation you have the person get the pathogen and have to fight it off for themselves, produce memory cells which are going to be there for the long run. Okay, so antibodies are rapidly broken down, and that is actually true. There are no memory cells produced, so C is wrong. Antigens are rapidly broken down. I mean, that is not the reason we get memory, long-term immunity. Okay, phagocytes soon die. Again, not the point. Antibodies are the ones being broken down, because antibodies are the ones being administered. Which row is correct for control and prevention methods for each disease? Well, let's see. So TB, control and prevention, is vaccination, chlorination of water, contact tracing, destruction of a vector. I have to say vaccination and contact tracing. Chlorination of water would be more effective for waterborne diseases, like in the case of cholera. Okay, and destruction of a vector, obviously there's not a vector, an animal or some organism that transfers the pathogen from one person to another, like in the case of malaria, and that's why we need to destroy the vector for malaria, chlorinate the water, and specifically do contact tracing. Vaccination isn't as effective for tuberculosis because there are many requirements to keep the vaccine working and safe, whereas contact tracing allows people to be, someone who's in their infective stage to be isolated, and so that basically allows us to minimize the cases of tuberculosis. Peptidoglycan is stained purple by a chemical crystal violet. Which cells would stain purple in the presence of crystal violet? Okay. Peptidoglycan is stained purple, meaning we need a bacterial cell. Is it palisade mesophyll cells? It's a plant cell. Vibrio cholerae, of course, Vibrio cholerae are bacteria. Plasmodium falciparum. Plasmodium falciparum is a species of Protoctist, therefore that is wrong. Okay, and endothelial cells, okay, therefore that's B. And that's it for this video. I hope this video was useful for you. If it was, make sure to like, subscribe, and share this video with someone who might need it. And that's it for this video. I'm gonna see you guys in the next one. Goodbye.